Hello and welcome back. My name is Matthew Belzer. I'm your A&P2 instructor, and if you're watching this, you're probably in my A&P2 class. Welcome to the lecture over fluid and electrolyte balance. I may break this lecture into a couple of different parts. Here are your learning objectives. I'd highly recommend printing out your lecture handout and having that out and available so you can kind of follow along with what we're doing today. Now, there are three main themes to this lecture: water balance, salt balance, pH balance. So we're going to go over water balance first, then salt balance, then pH balance, and that's how the lecture is going to be structured. In order to understand water balance, it's kind of important to understand the relative distribution of electrolytes in our different body compartments. So here we have blood plasma, and here we have interstitial fluid. These are both collectively referred to as extracellular fluids. So the interstitial fluid and the blood plasma are forms of extracellular fluid. You have about 3 liters of blood plasma, 5 liters of whole blood, about 3 liters of blood plasma, and you have about 12 liters of interstitial fluid. So we have way more interstitial fluid than we have blood plasma. Then you have intracellular fluid. Your body is composed of about 200 trillion cells, and the intracellular fluid is the fluid inside of those cells. And when you look at the relative distribution of electrolytes or ions between these different body compartments, you see a trend start to emerge. Number one, the system is never in equilibrium. Never, ever in equilibrium. Living systems are always in what's called chemical disequilibrium, and we expend a lot of energy maintaining this chemical disequilibrium, meaning that there are different concentrations of electrolytes in our different body compartments, or solutes in our different body compartments. When we look at this in general, this graph or this particular diagram should make a lot of sense. We look inside the cell and you go, wow, there's a really high concentration of potassium. Well, we've talked about the sodium-potassium pump in detail. Every cell has tons of sodium-potassium pumps, and for every cycle, 2 potassiums are moved into the cell. So it makes sense that potassium would start to accumulate in this cell, right? We're creating that gradient for potassium, so when potassium channels open, potassium rushes out of the cell, and you have things like repolarization. You also have a really high concentration of proteins inside the cell because the product of gene expression is proteins. And those proteins are what actually carry the negative charge inside the cell that allows the sodium-potassium pump to create a net negative membrane potential. Outside the cell, you'll notice there are much higher concentrations of sodium chloride and bicarbonate, right? Very low concentrations of potassium. Well, when you think about the sodium-potassium pump, right? 3 sodiums out, 2 potassiums in. So it goes to follow that sodium's going to build up outside the cell. It's going to suck chloride with it. When you move salt, you move water. And the concentrations of sodium chloride in the interstitial fluid and the blood plasma are relatively equal. The difference between interstitial fluid and blood plasma with respect to solute concentration really comes with proteins. Plasma proteins don't get pushed across the capillary wall. They remain in the blood. And the most notable of the plasma proteins that accounts for a significant percentage of this little bar right here are albumins. And that's why the blood plasma has a higher what's called blood colloid osmotic pressure than the interstitial fluid, and that's why it will, at the venous end of a capillary, reabsorb or suck water back from the interstitial fluid to the blood plasma, because water follows solute, specifically non-penetrant solute. So when you think about what maintains this chemical disequilibrium, be familiar with which ions you find in higher concentrations in the ICF and the ECF, and know that this chemical disequilibrium is impacted or established by ultimately the sodium-potassium pump. Now, why do we care? Because osmolarity and tonicity affect cell size and volume. So you're going to get questions like this on the exam. What will happen to the ICF volume, the intracellular volume, if ECF non-penetrating solute concentrations increase dramatically? What will happen to the ICF volume if ECF non-penetrating solute concentrations decrease dramatically? Now, with that framework in mind, let's think about what influences the movement of fluid between the different body compartments, between the plasma and the interstitial fluid, and between the interstitial fluid and the intracellular fluid. What are the primary drivers? Well, the primary driver of the movement of fluid between the blood plasma and the interstitial fluid is hydrostatic pressure. That's the force that promotes what's called net filtration, the movement of plasma across the capillary walls into the surrounding space where it becomes interstitial fluid. Anything that increases net filtration pressure is going to increase the amount of fluid exchange at the level of capillaries, right? So we can talk about things that will increase that, and in fact, we'll make some predictions in just a minute. But that's the primary driver or influencer of shifts between the plasma and the interstitial fluid. On the flip token, the primary influencer between shifts that occur, meaning fluid movement between the interstitial fluid and the intracellular fluid, is osmolarity. And when you think about osmolarity, you were probably introduced to the concept of tonicity. They're not interchangeable terms, but it's a good framework to start thinking about it on. When you place cells into hypertonic solutions, right, because there's a higher concentration of non-penetrating solutes outside the cell than inside the cell, water follows non-penetrating solutes. Sodium chloride, or salt, is your most important non-penetrating solute, and that'll suck water out. And in hypertonic solutions, because water is moving from the ICF to the ECF, the cells shrivel or crenate. It's called plasmolysis or crenation. Crenation is just the shriveling or shrinking of cells. In isotonic solutions, where the concentration of non-penetrating solutes, notably salt, is the same between the ECF and the ICF, you don't have any net movement of water, and isotonic solutions don't change the size and shape of cells. Hypotonic solutions, on the other hand, are when you have a really low concentration of non-penetrant solutes in the ECF and a higher concentration of non-penetrant solutes in the ICF. Water follows salt, water flows into the cell, and those cells can swell, and if it's hypotonic enough, they can eventually lyse, right? Hemolysis is just the breaking open of red blood cells. So we care about fluid and electrolyte balance because it influences the size and shape of cells. Here we have another depiction of this. Isotonic, same concentration of non-penetrant solute represented by the black dot in the inside of the cell and the outside of the cell, so you don't get any net movement of water. Hypotonic, right? Very low concentrations of non-penetrating solute in the ECF and higher concentrations of non-penetrating solute in the ICF, represented by those black dots. Water, right, moves from the ECF to the ICF down what's called an osmotic gradient. The cell swells and it can potentially burst if it's hypotonic enough. Hypertonic, much higher concentrations of non-penetrating solute in the ECF than the ICF. Water follows non-penetrating solute. Water follows salt and sugar. So it's going to suck water out of the cells, and the cells are going to shrivel or crenate. I know that these two images don't overlap exactly, but think about what this is telling you with respect to the concentration of non-penetrating solutes inside versus outside the cell. Because I could just ask you, if the concentration of non-penetrating solutes outside the cell is less than that inside the cell, what's going to happen to the cell? So you go, well, let's start thinking about that. One way to think about that is to give you an actual situation. So there's no difference between this question I'm about to ask you and the neuron question, right, that you have. It's just where are the fluid fluxes occurring and how is that impacting different body structures. So let's say there's a situation in which the concentration of your extracellular fluid, specifically your interstitial fluid, went down. What can produce that? Well, drinking excessive amounts of water without getting corresponding electrolyte input can decrease the concentration of electrolytes in your blood. Imagine putting a teaspoon of sugar into water, mixing it, and then pouring in more water. Is it going to get more concentrated or more dilute? Well, the more water you add without corresponding solute, it's going to get more dilute and more dilute. That's kind of one of the dangers of drinking too much water, right? You can actually dilute out your blood and you can create what's called a hypotonic condition. So in this case, the interstitial fluid is 0.45% and the intracellular fluid is 0.9%. These are little protein channels called aquaporins, meaning they allow the movement of water but not solute. The most important non-penetrating solute, meaning solutes that can't get across the cell membrane, is sodium chloride. And only non-penetrating solutes influence tonicity. In other words, water will always follow non-penetrating solute. Salt and sugar are two good examples of non-penetrating solutes. So you drink a bunch of water and the concentration of your interstitial fluid goes down, right? Well, if it goes down, how's that gonna impact the cell? Well, there's two ways to look at this. Water always follows salt. So water is gonna move from areas of low salt to areas of high salt, right? And water is gonna move from the outside to the inside of the cell. The cell is gonna swell, and if it was hypotonic enough, potentially burst, right? So that's a review from A&PII. Another way to look at this problem is if this is 0.45% sodium chloride in the interstitial fluid, it's 99.55% water. And the inside of the cell, if that's 0.9%, it's 99.1% water. Water will always flow from areas of high water potential to areas of low water potential down its concentration gradient, or its osmotic gradient, which is a concentration gradient for water. So let's look at another circumstance here. Let's say you put a red blood cell into this solution, 3% sodium chloride, which is an IV you run on people, right? And you go, okay, well, what's going to happen to the red blood cell? In which direction is water going to move? Is it going to move into the cell, or is it going to move out of the cell? So you can look at it as water follows salt, 0.9% sodium chloride and 3% sodium chloride. It's always going to follow the salt, so water is going to move from the ICF to the ECF, and it's going to get sucked out of the cell. But the reason that water does that, the reason that water follows solute, is it follows its osmotic gradient, which is the concentration gradient for water. So another way to look at tonicity is if this is 0.9% sodium chloride inside the cell, we assume the rest is water for the sake of this particular discussion with this solute. So it's 99.1% water. If this is 3% sodium chloride, then the solution is 97% water. Water will move from areas of what's called high water potential to areas of low water potential. So water follows an osmotic gradient. There's a gradient for water in which water moves from areas of high, what's called water potential, to areas of low water potential. That's why water follows solute. You run different IVs in different situations. So this is kind of forming a basis for what we think of as IV science. And again, we're reviewing some of the concepts of osmolarity and tonicity with a focus on tonicity because that's what influences the movement of fluids between the inside and the outside of the cell. So let's say we had a dude who's in the desert and hasn't had a drink of water in 24 hours, right? What's going to happen to the tonicity of the blood? Well, what's going to happen to the concentration of non-penetrant solute in the blood? Meaning, what's going to happen to salt concentrations in the blood? You become really dehydrated, you're losing more water than salt. Salt concentrations are going to go up. When salt concentrations go up, right, we've already seen water follows salt, so water is going to move from the intracellular fluid to the extracellular fluid. So when you get really dehydrated, you're actually sucking water out of your cells. And what's that going to do to the size and shape of these cells internally? It's going to cause them to shrivel or crenate, and they're not going to be able to function appropriately. So how would you correct something like intracellular dehydration? Intracellular dehydration can be caused by not having enough water for a prolonged period of time. It can be caused by having too much salt. Let's say you intake you got a little kid and they eat a bunch of salt, because sometimes kids do that. Well, the salt concentrations in their blood spike and suck water out of cells. Diabetic ketoacidosis. Get the buildup of glucose and ketone bodies outside the cell that sucks water out of the cell. In all of those situations that produce what's called intracellular dehydration, you're going to want to run a hypotonic solution. And the most common hypotonic solution to run is what's called a half saline solution, or a 0.45% sodium chloride solution. That's a very common solution to run on people who are suffering from intracellular dehydration. Why? Well, if water is being sucked out of cells, you want to move water back into cells. So you apply a hypotonic solution. On the flip token, when would you run a hypertonic solution? Well, if somebody is suffering edema, like maybe in the case of the question that I asked you about the neurons, right? Edema is a buildup of fluid in your tissues. And how do you suck water out of tissues? Well, the only way that you can actually physically move water, because you can't make a bunch of little pumps to do it, is you rely on osmotic gradients. You go, okay, we have a bunch of water fluid collecting, let's say, in the brain, and we want to suck that fluid out. Let's run a hypertonic solution so we can suck the fluid out of the cells there, move it back into the blood, and make it easier for the body to handle. Isotonic solutions, like what's called physiological saline, are run in all sorts of situations. Mild forms of dehydration to increase blood pressure, because when you increase blood volume, you increase blood pressure. That's why somebody who's like hemorrhaging, they might say 2 liters of saline wide open just to increase their blood pressure before they know anything about the blood products or anything. So you run different IVs in different circumstances. Again, really think about that neuron question in learning objective 2. What's the change in the solution? In which direction is water going to flow? What's that going to do to the neurons in the brain? And why is that so dangerous? So when we talk about integrated responses are responses in which more than one organ system is responsible for controlling a physiological variable, like blood volume or blood pressure or heart rate or whatever it is. A good example of integrated responses are blood volume and blood pressure, the regulation of blood volume and blood pressure. So let's look at what that actually means. Let's say that blood volume drops. Well, if blood volume drops, blood pressure will drop. And when blood pressure drops, that's detected by what are called baroreceptors, pressure receptors, in places like the carotid arteries and the aortic arch. Now, that triggers reflexes that will influence the cardiovascular system. And the cardiovascular system will go, ooh, we need to increase cardiac output and vasoconstrict blood vessels because we want to bring blood pressure back up. It'll also influence behavioral circuits, and you'll start to feel thirsty. You'll drink water, blood volume will go up, blood pressure will go up. So now we have the cardiovascular system, the nervous system, and the urinary system working together. When blood volume and blood pressure drop, you release hormones like antidiuretic hormone that's going to tell the kidneys, hey, we need to reabsorb as much water as we possibly can, decrease the amount of urination because we want to bring blood volume and blood pressure back up. So you have multiple organ systems working together, right, to maintain blood volume and blood pressure. On the flip token, blood volume and blood pressure gets too high. Well, when it gets too high, right, the baroreceptors and the aortic arch and carotid bodies go, oh, cardiovascular system, those are way too high, right? So you decrease cardiac output, parasympathetic nervous system activates, vasodilation, that's going to lower blood pressure. The kidneys, you stop giving antidiuretic hormone and you say, hey, kidneys, we need to get rid of more salt and water. So let's start urinating more because we want to bring that blood volume back down. Different systems working together to mediate a single effect, in this case, blood volume. So now we're going to look at water balance. We have a little bit of a framework to start thinking about water balance. Any type of mass balance problem really revolves around, let's see here, jumping the gun. Any type of mass balance problem really revolves around input and output. So where do we get water? In food and drink, about 2.2 liters a day. We also have metabolic water production because water is the byproduct of burning glucose in the presence of oxygen in order to create energy in the form of ATP. So that's what metabolic water production is. Collectively, that's input. We have about 2.5 liters of water input a day. If we're in mass balance, our input should be equal to our output. So we lose water through the skin. Even if you're not sweating, you lose a little water through evaporation. It's called insensible water loss. Every time you exhale, you lose a little bit of water. We lose about 0.9 liters a day that way. In the urine, we lose about 1.5 liters of water a day. In the feces, about 0.1 liters of water a day, right? So if water input and water output are roughly equal, you're in a good situation. All mass balance revolves around that. What's the input? What's the output? Caloric mass balance. What's your caloric input? What's your caloric output? Protein mass balance. What's your amino acid input? What's your amino acid output? Right there, mass balance is just input subtract output. And ideally, in a healthy system, we kind of want it close to zero, right? Unless you're trying to do different things and all. I won't get into that. So let's look at different conditions that influence mass balance in the body, and let's see if we can explain what would happen, right? So now let's say we're doing this. 2.5, 3.3. Yeah, okay. So now let's say we're doing this. So input and output are now not balanced, and what you see is you're losing a lot more water in the fecal matter, about 0.9 liters a day. That's a lot to lose in the fecal matter. So what's a condition in which you would be losing more water in the fecal matter? Well, it could be something like diarrhea, right? You could have diarrhea. So why is diarrhea so dangerous? It throws off what are called balances. You have water input and you have water loss. It's why you need to be very careful about staying hydrated if you have diarrhea. So now we're losing about 0.8 liters of water in that day as a consequence of the diarrhea. If we're losing water, what's going to happen to the concentration of solutes in the blood plasma? They're going to go up. The body's going to interpret that as dehydration because an increase in the concentration of salt is what the body is actually monitoring to say, hey, I'm dehydrated. What's going to happen to blood volume? Blood volume is going to decrease. What's going to happen to blood pressure? Blood pressure is going to decrease. So when you think about this, think about the different conditions that can be produced, like from diarrhea. And what's that going to do to the size and shape of the cell? So I'm going to give you a mass balance problem in which you have numbers here, and I'm just saying, is this person overly dehydrated or dehydrated? And you can look at the different conditions in Learning Objective 6 and see whether you can kind of put together what I'm trying to get at there. So now we go to Learning Objective 5, looking at fluids, major fluid compartments in the body. Intracellular fluid, right? You have about 25 liters of intracellular fluid. It is where the majority of the water in your body is held. So exchange between the interstitial fluid and the intracellular fluid is really important. We have about 12 liters of interstitial fluid and we have about 3 liters of blood plasma. So even though it doesn't seem like it, we have the least amount of blood plasma. Think of an ordering question there. From greatest to least, which of these compartments contain more water? And that's our total body water. Now, one of the labs we would have done is we would have had people drink hypertonic drinks like V8, hypotonic drinks like water, and isotonic drinks like Gatorade. And we would have looked at the direction of fluid flow in the body. Now, we're not doing that, but let's see if we can make some predictions based on what we know. You take in an isotonic load. Classic isotonic load is Gatorade. Gatorade has the same concentration as your blood plasma and your intracellular fluid. So when you drink Gatorade, blood volume is going to go up, and it's going to force blood pressure to go up because when blood volume goes up, blood pressure goes up. When blood pressure goes up, you get more exchange between the plasma and the interstitial fluid because that's the primary driver of exchange between these two compartments. But you're not going to get much exchange between the interstitial fluid and the intracellular fluid because you just drank an isotonic solution, so you're not going to get the fluid fluxes that occur here. On the flip token, if you drink pure water, right? Blood volume is going to go up, and therefore blood pressure is going to go up. Fluid is going to increase. The movement of fluid between the plasma and the interstitial is going to increase as a consequence of volume changes, volume and pressure changes, right? And if you have a hypotonic load, that's going to start decreasing the amount of non-penetrating solutes in the interstitial fluid. And what that's ultimately going to do is it's going to force water into cells, and that's a way to kind of plump cells back up. So think about hypertonic load. Let's say you drink a bunch of V8. Well, the concentration of your the volume of your blood plasma will increase, therefore the pressure will increase. You'll get a transition between the plasma and the interstitial fluid, but because that's a hypertonic load, the concentration of non-penetrating solutes in the interstitial fluid increases, and it'll start sucking water out of cells, right? So it's interesting how different fluid loads can impact things. So when you're working out, let's say you have an excessive loss of water from the extracellular fluid. Extracellular fluid osmotic pressure rises. Wherever there's a higher concentration of salt, you have a higher osmotic pressure. So if you're losing water during exercise, the concentration of non-penetrant solute, the salt concentration in the ECF is going to increase, right? Wherever there's a higher concentration of non-penetrating solute, salt and sugar, right? That's always going to have the higher osmotic pressure. And what that osmotic pressure is going to do is it's going to force water out of the cells. So water is going to move from the ICF to the ECF, and the cells are going to start to shrivel and crenate. In order to correct that, you have to drink water. So if you drink water, you're going to decrease the concentration of non-penetrating solutes in the ECF. If you decrease the concentration of non-penetrating solutes in the ECF, you create a hypotonic solution and you force water into cells. The danger in both of these is going to extremes, either with water loss or with water gain. Again, think about that neuron question and think about what the effects of that would be. Now, when we think about water balance, we have to come back to the hypothalamus. When you look at graphs like this, ECF osmolality increases. The way the body detects dehydration is it looks at the concentration of salt. When the concentration of salt in extracellular fluids like your blood plasma increases, that's detected by what are called osmoreceptors in the hypothalamus. Osmoreceptors in the hypothalamus do a couple of different things. The first thing they do is they activate thirst centers, and then we start to feel thirsty, and we will drink water in response to that. Water gets absorbed from the GI tract, and when we're drinking water, that's going to lower the concentration of the blood plasma and bring us back to normal. Additionally, when the blood volume drops, that activates a separate system that we're going to talk about when we get into salt balance. The big thing here when you're thinking about dehydration is to focus your attention on antidiuretic hormone. And we talked about antidiuretic hormone when we were going over the urinary lecture just a little bit ago, but I want to bring up antidiuretic hormone again. So, concentration of salt increases. The hypothalamus detects that as dehydration. So the hypothalamus, because it can come into contact with blood, goes, ooh, salt concentrations have increased, right? I better tell the posterior pituitary gland to release a hormone called antidiuretic hormone. Antidiuretic hormone targets the collecting duct of nephrons in the kidney. And in the presence of antidiuretic hormone, the cells of the collecting duct insert aquaporins, and that allows them to reabsorb water. As water is being reabsorbed, the concentration of the blood plasma will go down, the plasma volume will go up. You're not going to urinate very much, right? Because you're reabsorbing most of that water, so you're going to have a small amount of very concentrated urine. This is our response to things like dehydration. So, let's think about this from beginning to end. The antidiuretic hormone, aka arginine vasopressin pathway. ADH and AVP are different words for the same thing, and different people use the conventions based on what their academic background is. You get dehydrated. The concentration of salt in your blood plasma goes up. That's detected by osmoreceptors in the hypothalamus because the hypothalamus can come into contact with blood. Those osmoreceptors activate two pathways. First, they activate the thirst pathway. So when you're dehydrated, you actually feel thirsty, and that drives you to intake water, right? That's our physiological cue to intake water. So behaviorally, we get thirsty, we're going to drink water. Physiologically, the osmoreceptors tell the posterior pituitary gland to release a hormone called antidiuretic hormone or arginine vasopressin. So the stimulus for the release of antidiuretic hormone is an increase in the concentration of salt in the blood. The receptor are the osmoreceptors in the hypothalamus. The control center is the hypothalamus. What that does is it activates two pathways. The first is behavioral, and we get thirsty and we start drinking water. The second is hormonal, and the target of those hormones are the collecting duct of the kidneys. And when antidiuretic hormone binds to the cells in the collecting duct of the kidneys, it triggers the insertion of aquaporins, which allow the collecting ducts to reabsorb water. As we reabsorb water, the concentration of salt in the blood is going to go down, meaning that this is a negative feedback system. Blood volume is going to go up, blood pressure is going to go up, and when you're dehydrated, you produce a small amount of very dilute, very concentrated urine because you're reabsorbing every last stitch of water that you possibly can. So again, you don't have to know the chemical formula of arginine vasopressin, but I do expect you to be familiar with this pathway with a focus on osmolarity or salt concentrations, because osmolarity is just a measure of concentration. So when we look at this, we go, okay, salt concentrations increase. That's our stimulus. That's detected by osmoreceptors in the hypothalamus. Those osmoreceptors in the hypothalamus activate our thirst centers and change our behavior, so we start drinking water. They also, activate hormonal pathways. So the hypothalamus tells the posterior pituitary gland, hey, release antidiuretic hormone, aka arginine vasopressin. Antidiuretic hormone then travels through the blood to the collecting ducts in the kidneys where it binds to receptors and it triggers the insertion of aquaporins. When aquaporins are inserted, water starts to be reabsorbed. So that water that gets reabsorbed is going to bring the concentration of salt in the blood back down. It's going to jack up our blood volume again so we can maintain our blood pressure. And when you're dehydrated, you produce very small amounts of concentrated urine. Think about this image from the previous section. So I could ask you on something like this, which of the following would be true? Check all that apply. Well, here we're looking at the collecting duct of the nephrons in the kidney, and these are representative of many, many nephrons, but we're looking at total physiological condition. So if you see the concentration of the filtrate that enters into the collecting duct is low, and it starts getting more and more concentrated as a consequence of reabsorbing water along the collecting duct, there's only one explanation for that. The collecting duct cannot reabsorb water in the absence of antidiuretic hormone. If the collecting duct is reabsorbing water, it means antidiuretic hormone concentrations in the blood are high. And what would increase the concentration of antidiuretic hormone? You get dehydrated, salt concentrations increase in the blood. That's detected by the hypothalamus, specifically the osmoreceptors in the hypothalamus. The hypothalamus is the control center. It processes that information. It activates different systems. Behaviorally, we get thirsty and we start to drink water in order to dilute out our blood plasma and bring the concentration back down, also to bring our blood volume and our blood pressure back up. Additionally, those osmoreceptors trigger the posterior pituitary gland to start releasing antidiuretic hormone or arginine vasopressin. Antidiuretic hormone is necessary for the reabsorption of water along the collecting duct in the kidney. So if water is being reabsorbed along the collecting duct in the kidney, all of those solutes, remain in the collecting duct as part of the filtrate, and we are reabsorbing water, right? If you are reabsorbing water along the collecting duct, it means you are dehydrated and your body is trying to retain as much water as possible. It only does that in response to antidiuretic hormone. And in this situation, we're going to produce a very small amount of super concentrated urine. And that urine would be like dark yellow or even brown, depending on how dehydrated you are. So you know that this person over here is dehydrated. On the flip token, if somebody is well hydrated, we don't need to reabsorb water in the kidneys, so the kidney is going to flush that water out. So you have the filtrate flowing through the collecting duct, but there's no absorption of water. Well, if we're not reabsorbing water in the collecting duct, it means that the concentration of salt in the blood is low. The hypothalamus goes, okay, low salt concentrations, no need to reabsorb water because we're well hydrated. No antidiuretic hormone is released. In the absence of antidiuretic hormone, the collecting ducts do not reabsorb water. If they're not reabsorbing water, you produce a large amount of very dilute urine. This person over here would be well hydrated and their urine would be very light yellow, clear, kind of straw color, right? So you can tell a lot by looking about somebody's physiological condition by looking at the status of the collecting duct. And we've gone over a ton of this. Now, the final thing I'm going to hit on again. Use the image below to fill out the table. ADA concentrations, blood plasma, hydration status, urine volume, urine color. You should be able to answer those things based on the framework that I just gave you. Another really important set of structures in regulating salt and water balance in the kidneys are the descending and ascending limbs of the loop of Henle and the descending and ascending vasa recta. Now, we talked about this countercurrent exchange mechanism when we were going over urinary physiology, so I'm not going to beat it to death, but I am going to review it again for you. So when we were doing urinary physiology, we looked at every segment of the nephron, and we looked at what make those segments of the nephron unique. Now, the vasa recta are the capillary networks you find in the renal medulla, right? Deep in the kidney. And the vasa recta interact with the loops of Henle. One of the most important things to understand here is that blood flow in the vasa recta will always move in the opposite direction of filtrate flow in the loops of Henle. So when you look at this, this is what's called countercurrent because blood and filtrate are moving in opposite directions. Exchange because there's an exchange between the nephron and the blood. So when you look at the ascending limb of the loop of Henle, you look at the ascending limb of the loop of Henle. In the cells lining the ascending limb of the loop of Henle, you have a unique protein called the sodium-potassium 2-chloride cotransporter. The sodium-potassium 2-chloride cotransporter. So in the ascending limb of the loop of Henle, we reabsorb salt and only salt. And where are we reabsorbing that salt? We're reabsorbing it into the blood. So if we're reabsorbing salt and only salt into the blood, the descending vasa recta, the blood is going to become more and more concentrated. On the flip token, as we move salt out of the filtrate is going to become more and more dilute. So why do we make the blood in the descending vasa recta so concentrated? It's because we want to create an osmotic gradient that we can use to suck water up, because you first have to move salt before you move water. So the ascending limb of the loop of Henle, because it has these sodium-potassium 2-chloride cotransporters, creates an osmotic gradient, meaning that it makes the blood super duper salty. Then that blood in the ascending vasa recta contacts the descending limb of the loop of Henle. Water follows solute in the descending limb of the loop of Henle. Those are the cells that always have aquaporins. So if there's an osmotic gradient, right, in other words, water following salt that promotes the movement of water from the filtrate into the blood, and there is, we're going to get the reabsorption of water along the descending limb of the loop of Henle. So the descending limb of the loop of Henle is capable of reabsorbing water and only water because it uses the osmotic gradient that's generated by the ascending limb of the loop of Henle, which reabsorbs salt and only salt. So the ascending loop of the limb of Henle creates an osmotic gradient, and the descending limb of the loop of Henle uses that osmotic gradient to reabsorb water. The longer the limb of the loop of Henle, the more water reabsorption can occur. Water reabsorption isn't perfect, so it's fine-tuned in the collecting duct. This process is always going to be taking place. One of the questions that I always ask are, when you think about blood pressure medications such as furosemide, for example, well, those are what are called loop diuretics, and the way that furosemide or loop diuretics work is they work by inhibiting the activity of the sodium-potassium 2-chloride cotransporter in the ascending limb of the loop of Henle, and that's why they're called loop diuretics. If you inhibit the activity of the sodium-potassium 2-chloride cotransporter, you can't reabsorb salt. If you can't reabsorb salt, you can't reabsorb water. If you're not reabsorbing salt or water, all of that salt and water remains in the filtrate and ultimately becomes part of the urine. Your urinary output goes up, and as urinary output goes up, meaning we're getting rid of more salt and water in the urine, we're literally just urinating more, blood volume goes down and blood pressure goes down, and that's why loop diuretics are such an effective treatment for things like high blood pressure. So that brings us to the second part of what we're going over today, which is salt balance. So when you think about salt balance, there's a really important system that we are going to go over that plays just an extraordinarily important role in salt balance. Now, why are high sodium diets linked to things like high blood pressure? Why, when people have high blood pressure, do doctors suggest restricting sodium chloride intake? Well, let's look at how that affects the systems we just talked about. You ingest salt. Let's say we ingest salt without corresponding water. Well, if you're eating salt without corresponding water, the concentration of your blood is going to increase because you're going to be absorbing that salt, right? Let's say you're at a bar and you're just eating pretzels or whatever. The concentration of salt in the blood is going to go up. When the concentration of salt, or the osmolarity of the blood plasma, goes up, that's detected by the hypothalamus, specifically osmoreceptors. And those osmoreceptors go, oh, let's activate thirst centers so we get thirsty and start drinking water, right? It's one of the reasons they put pretzels at bars, to make you thirsty. 2, let's start releasing antidiuretic hormone. So as thirst increases, water intake increases, and as water intake increases, you're going to lower the concentration of the extracellular fluid, but you're going to jack up blood volume and therefore jack up blood pressure, right? Additionally, if you're releasing antidiuretic hormone in response to salty food, it's going to trigger more reabsorption of water along the kidneys. The kidneys are going to reabsorb water, and you're going to jack up your blood volume and your blood pressure, because the two are intricately kind of intertwined. So that's what I'm getting at with briefly explain why a diet that's high in sodium can increase blood pressure and is therefore contraindicated in patients with hypertension. That's exactly what I'm talking about in that particular question. Now, one of the primary mediators of salt balance in the body is a hormone called aldosterone. There are very few hormones you would immediately notice or die without. Antidiuretic hormone is one of those that you're going to die if you don't have it because you're going to lose too much water. Aldosterone is another hormone that you're going to die if you don't have it, right? It will ultimately lead to your death. So what we are going to look at is what's called the renin-angiotensin-aldosterone system or pathway. So on your exam, you're going to get a question that says, hey, Billy's walking through the desert and he hasn't had a drink of water in a prolonged period of time. Predict what's going to happen to these parameters and explain the body's response to that. There we're talking about the antidiuretic hormone pathway, which is a primary mediator of water balance, right? Here we are talking about a pathway called the renin-angiotensin-aldosterone system. And when you think about the stimulus right? For the renin-angiotensin-aldosterone system, there are 3 stimuli that activate this system. So it's saying, what are the 3 stimuli for the activation of the RAAS system? Stimuli number 1, and the most important by a long shot, is an increase in the concentration of potassium in the blood. Potassium is really, really dangerous. If potassium concentrations increase too much, it can prevent cardiac muscle cells from being able to repolarize, and it will stop your heart. So the basis of lethal injection is just a big bolus of potassium chloride that ultimately stops the heart because it prevents cardiac muscle cells from repolarizing because there's no potassium gradient. In other words, fluctuations in potassium are really, really dangerous. Gets too high, it'll stop your heart. Gets too low, it will paralyze you. So potassium concentrations in the blood are really important to maintain. The primary stimulus that activates this, the strongest activator of this pathway, is an increase in potassium. That's followed by a decrease in blood pressure. A decrease in blood pressure will also activate this system, or a decrease in the concentration of sodium chloride in the blood will also activate this system. Now, I'm going to focus on this particular graph right here because I think this graph does a really good job at summarizing exactly what's happening. So when you're looking at your handout, let's go to step 1. That blood pressure cuff is measuring blood pressure. There are 3 stimuli that can activate this pathway. On your handout, it's a decrease in blood pressure. So it's a decrease in blood pressure. But an increase in blood potassium, right, is the stronger stimulator, or a decrease in blood sodium. All 3 of those can activate this pathway. So let's say that blood pressure drops. Well, when blood pressure drops, that's detected by that system we talked about called the juxtaglomerular apparatus in the kidneys. And the cells of the juxtaglomerular apparatus, in response to that, release an enzyme called renin. Renin travels through the blood to the liver, where it converts angiotensinogen. Remember, agens are always inactive. It converts angiotensinogen into an active angiotensin 1. Angiotensin 1 then goes to the lungs, where it's converted to angiotensin 2 by what's called angiotensin converting enzyme, ACE. Angiotensin 2 is then very physiologically active. So it's going to activate the sympathetic nervous system. And when your sympathetic nervous system activates, your heart rate's going to increase and your blood vessels are going to vasoconstrict, and that's going to bring blood pressure back up, right? Second thing that I want you to know that angiotensin 2 does is it triggers the adrenal cortex, right, to release a hormone called aldosterone. And remember, we looked at the different layers. There, we're talking about the zona glomerulosa. We get the release of aldosterone. Aldosterone will travel to the principal cells in the collecting ducts of the kidneys, the same cells that antidiuretic hormone works on, and they'll bind to aldosterone receptors. When aldosterone influences the collecting duct, what it will do is it will trigger a reabsorption of sodium and a secretion of potassium. So sodium and chloride get reabsorbed and potassium gets secreted and eliminated in the urine. Makes sense that potassium concentrations would also activate this pathway. The final thing that I want you to know angiotensin 2 does is it triggers the posterior pituitary gland to release antidiuretic hormone, because if we're reabsorbing more sodium chloride, we want the corresponding water reabsorption to bring our blood volume and therefore our blood pressure back up. You will 100% be asked a detailed question on the renin-angiotensin-aldosterone system. So I am going to go over it again, even though I've already gone over it once, and I am going to bring up specific things to think about at each step. Let's now use an increase in potassium. So you eat a bunch of bananas and the potassium levels in your blood spike, and that can be really dangerous, so your body needs to start getting rid of potassium. Stimulus for the juxtaglomerular apparatus to produce renin. Increased potassium, decreased blood pressure, decreased sodium. In this case, we're talking about increased potassium. Really think about that on the exam, because you're going to have to trace out this pathway from beginning to end. So in response to increased potassium, the juxtaglomerular apparatus produces renin. Renin travels to the liver, where it converts angiotensinogen to angiotensin 1. Angiotensin 1 travels to the lungs, where angiotensin converting enzyme, or ACE, converts angiotensin 1 to angiotensin 2. Angiotensin 2 activates the sympathetic nervous system, which will bring up blood pressure, heart rate, and it'll trigger vasoconstriction of blood vessels, so it'll bring up blood pressure. It also triggers the adrenal cortex, specifically the zona glomerulosa, to release aldosterone. Aldosterone is called a mineralocorticoid, and what aldosterone is going to do, and this is the primary effect that angiotensin 2 has, is it triggers the release of aldosterone. Aldosterone is going to go to the collecting ducts in the kidney, and it's going to trigger the reabsorption of salt and the secretion of potassium. Right? So we're going to reabsorb sodium chloride, and we're going to start getting rid of potassium. So when you think about this pathway, when aldosterone is released, the concentration of potassium in the blood is going to go down, because we're moving potassium from the blood into the filtrate, and we're eventually going to get rid of it in the urine. The concentration of potassium in the urine is going to go up. Because potassium and sodium are moving in opposite directions, in response to aldosterone, sodium levels in the blood are going to go up, and sodium levels in the urine are going to go down. When we reabsorb sodium chloride, because angiotensin 2 also triggers the release of antidiuretic hormone, we're also going to reabsorb water. When you reabsorb water, blood volume goes up, blood pressure goes up. If you're reabsorbing water, you're not going to have as much filtrate turning into urine, so urinary output is going to go down, meaning you're going to produce less urine in response to the activation of the RAAS system. This is your primary mediator of salt balance. Another target of blood pressure medications are what are called ACE inhibitors. ACE inhibitors stand for angiotensin converting enzyme inhibitors. Angiotensin converting enzyme inhibitors prevent the conversion from angiotensin 1 to angiotensin 2, and because all of the actions of angiotensin 2 are going to increase blood pressure, ACE inhibitors are effective medications for decreasing blood pressure that are predicated upon an understanding of this system. Now, why ACE inhibitors? It's because that's what was discovered and then tested and trialed and brought through the phase 1 through phase 4 clinical trials. It's not the only target. Let's say you were a graduate student and you found a chemical compound that inhibited the activity of renin, right? And it had a safer profile. You would definitely want to have the intellectual property rights to that, because that could just as effectively treat blood pressure, high blood pressure, right? So knowing this system from top to bottom, if I give you anything that says potassium increases or blood pressure decreases, right? I'm really asking you, can you walk me through the RAAS system? You need to be able to walk me through this from beginning to end. You will 100% get a question about water balance, specifically the antidiuretic hormone systems. You'll 100% get a question about salt balance, specifically the RAAS systems. The most profound stimulator of the RAAS system is an increase in potassium. So anything that talks about fluctuations in potassium on the exam, especially on the short answer questions, immediately draw your mind to the renin, angiotensin, aldosterone systems, because that's probably what I'm talking about, are the RAAS systems. But a decrease in blood pressure or a decrease in blood sodium can also do this. So potassium concentrations increase or blood pressure decreases. That stimulates the juxtaglomerular apparatus to activate the RAAS system. Renin is released, Renin converts angiotensinogen to angiotensin 1. That happens in the liver. Angiotensin 1 is converted into angiotensin 2 by angiotensin converting enzymes, or ACE enzymes, in the lungs. Angiotensin 2 then travels to different regions where it has its impacts. One, it upregulates the sympathetic nervous system, which is going to jack up heart rate and constrict blood vessels. That's going to increase blood pressure. Two, it's going to trigger the adrenal cortex to release the hormone aldosterone. That's its primary effect, angiotensin 2's primary effect. Aldosterone is going to travel to the collecting duct, right? Specifically, it's going to bind to receptors in the principal cells of the collecting duct, and it is going to trigger the secretion of potassium, because if potassium concentrations are high, that could stop your heart. So we get rid of potassium in the urine, right? So blood potassium will go down and urine potassium will go up, and it triggers the reabsorption of sodium chloride. And because angiotensin 2 also triggers the release of antidiuretic hormone, we get salt and water reabsorption. So we reestablish the homeostatic blood plasma levels of sodium and potassium, right? This system is going to be asked from top to bottom. It's going to be asked from top to bottom. Now, when you look at the actual effects of aldosterone is a steroid hormone, meaning it's a lipid-soluble hormone. You should be able to recognize why that's a lipid-soluble hormone. It doesn't bind to an extracellular receptor, it actually binds to an intracellular receptor. And that intracellular receptor is going to activate gene expression. So through the process of transcription and translation, we're going to get the production of new proteins. Among those new proteins are potassium channels on the luminal side of the principal cells in the collecting duct, and sodium-potassium pumps, right? So the sodium-potassium pump is going to move potassium out of the blood into the cells lining the collecting duct. It's going to move sodium from inside the cells lining the collecting duct back into the blood, because sodium and potassium are moving in opposite direction. These potassium channels are then going to allow potassium into the lumen, where it will join as part of the filtrate and ultimately be eliminated in the urine. We're also going to get the movement of sodium from the filtrate through sodium channels, through the activity of the potassium pump, sodium-potassium pump, back into the blood. So aldosterone is a steroid hormone that activates gene expression, and it causes the synthesis of these proteins from scratch, notably sodium-potassium pumps and potassium channels. Those are the mechanisms of action of aldosterone when they actually get to a cell. And that's why the concentration of potassium in the urine increases and the concentration of potassium in the blood decreases in response to aldosterone, because the strongest activator of the RAAS system is an increase in potassium levels. You see that on the short answer response, you're immediately thinking RAAS system, right? And it triggers the reabsorption of sodium, and what follows is chloride. When sodium chloride is being reabsorbed, water is also going to be reabsorbed, because we get that antidiuretic hormone that's being released in tandem. We reabsorb salt and water. It's going to reestablish both blood volume and blood concentrations. And those are the effects of aldosterone. Know that RAAS system from top to bottom. You will 100% get a question on the RAAS system. Now, just as a side note, whenever there's a hormone or a neurological mechanism that does one thing in the body, there's always something that does the opposite. So the hormone that does the opposite of aldosterone is a hormone produced by our atria. So our atria have endocrine aspects to them as well, the atria in the heart, called atrial natriuretic peptide. And it does the exact opposite of aldosterone. So whereas the RAAS system is activated by decreases in blood pressure or increases in blood potassium, this system that triggers the release of atrial natriuretic peptide is triggered by increases in blood pressure. So when blood pressure increases, it puts stretch on the wall of the atria. In response to that stretch on the wall of the atria start to release something called atrial natriuretic peptide, or ANP. ANP does the exact opposite of aldosterone. It triggers the secretion of sodium chloride. As sodium chloride moves into the filtrate and ultimately the urine, it sucks water. So we get rid, it'll produce a situation in which you are urinating more, and we get rid of more salt and water, which brings our blood volume and our blood pressure back down. It's called an antagonistic control mechanism. So when you're thinking about the atrial natriuretic peptide pathway, it's not going to be a primary focal point. You're not going to get a short answer question on it, but it does the exact opposite of aldosterone. So in responses to increased blood pressure, the atria stretch, the cells of the atria release a hormone called atrial natriuretic peptide. Atrial natriuretic peptide's primary effects are going to be decreasing salt and water reabsorption along the kidneys. When salt and water reabsorption decrease, the amount of urine you produce increases. You start peeing out your blood, essentially. That decreases blood volume and decreases blood pressure. So it does the exact opposite of what aldosterone does. The system you need to know in detail is the RAAS system. It's an incredibly important system in the maintenance of salt and water balance, and you would die very quickly without aldosterone. Now, the final activity we're going to go over is acid and base balance, pH balance. So with pH balance, remember what pH means. pH means power of hydrogen. So when you think about what an acid is, like hydrochloric acid, an acid is anything that when put into solution dissociates, meaning splits apart, to form free hydrogen ions. Strong acids, you get complete dissociation. So when you throw hydrochloric acid into water, every molecule of hydrochloric acid, right, will dissociate to form free hydrogen and chloride. As free hydrogen concentrations go up, pH goes down, making a system more acidic. Strong acids dissociate completely. Weak acids like carbonic acid, associated with the carbonic acid bicarbonate buffering system, don't dissociate completely, right? When you throw carbonic acid into water, right, some of it will dissociate to form free hydrogen, some of it will remain intact. That's why we refer to carbonic acid as being a weak acid. It's not going to produce as much free hydrogen in solution as a strong acid does, like hydrochloric acid. Now, when you're thinking about where acids can come from in the body, you have the breakdown products of proteins, which can release something called phosphoric acid. Anaerobic respiration can create lactic acid. The breakdown of fat can create ketone bodies, and those are ketone acids. Or the intakes of substances like Mountain Dew or Coca-Cola, which are really acidic. Acid-base balance is essentially handled in three ways. The most important are our chemical buffering systems, specifically the carbonic acid bicarbonate buffering system, which you need to know for this exam. You need to know cellular respiration, that equation, and you need to know the equation for the carbonic acid bicarbonate buffering system, because we're going to talk about that, and that's going to form the basis of justifications for answers you're going to have on the exam. Now, these buffering systems take on the overwhelming majority of acid-base balance, and they're tweaked by respiratory centers. So the respiratory system can contribute to these buffering systems, and it can increase or decrease pH by managing the amount of carbon dioxide we have. The urinary system, or the renal system, can also increase or decrease pH by handling hydrogen and bicarbonate directly. Now, respiratory compensation is very quick, like 1 to 3 minutes. Renal compensation can take hours or days. That's going to make a lot of sense in just a moment. The carbonic acid bicarbonate buffering system. You need to know this. This is the most important buffering system in the body. So let's go over what's actually happening and how these acids are produced. When you burn glucose in the presence of oxygen in order to make energy in the form of ATP, the byproducts are carbon dioxide and water. When carbon dioxide and water react in the presence of an enzyme called carbonic anhydrase, they immediately form this molecule called carbonic acid. Carbonic acid is a weak acid. And when carbonic acid dissociates, it forms free hydrogen and bicarbonate. Bicarbonate, this HCO subscript 3 with a negative charge, is a weak base. Right? So you have a weak acid and a weak base. All buffering systems are made from weak acids and weak bases. A strong acid will never be a good buffer because it will dissociate completely. A strong base will never be a good buffer. You need a weak acid and a weak base. The reason why is these reactions are reversible. So these reactions can be pushed to the left or to the right depending on body conditions. Right? So in other words, let's say that the body becomes too acidic, meaning that the concentration of free hydrogen increases. By the way, blood pH, which is something you absolutely need to know, is between 7.35 and 7.45. Anything below 7.35 is a condition called acidosis. Quite a few people were confused about high and low or increases and decreases on pH. If that's something that's confusing you, review pH. So when hydrogen ion concentrations go up, pH goes down, the system becomes more acidic. When hydrogen ion concentrations go down, pH goes up and the system becomes more basic. So let's say that this system, your blood, becomes too acidic. It means that the concentration of hydrogen ions is high. So if that becomes too acidic, what buffering systems do is they're designed to maintain relatively stable pHs. If hydrogen ion concentrations increase, it will drive the reaction to the left, in which carbonic acid will be reformed and CO2 and H2O will be formed, right, in order to remove free hydrogen from the system. On the flip token, if free hydrogen levels drop, pH goes up. If it becomes too alkaline, the system will shift to the right. So we get more free hydrogen. So think about shifts to the left and shifts to the right when you're talking about the carbonic acid bicarbonate buffering system. Make sure you write that out when you are giving me a justification of how the body is going to respond to different changes with respect to pH. The most important thing for you guys to know is this buffering system and what influences this buffering system. So the respiratory system influences pH by controlling the levels of carbon dioxide. The respiratory system influences pH by controlling the levels of carbon dioxide. When pH gets too low or too acidic, right, in a healthy system, respiratory rate will increase in order to get rid of carbon dioxide because there's a link between carbon dioxide and free hydrogen. You get rid of CO2, you get rid of free hydrogen. If the system becomes too alkaline, respiratory rate will decrease, which is called hypoventilation. So increase is hyperventilation, decrease is hypoventilation, in order to retain CO2 to push the reaction in this way to trigger the release of more free hydrogen. So that's how the respiratory system manages pH. This is really important. The renal system manages pH by managing the amount of hydrogen and bicarbonate directly. So if the system becomes too acidic, the kidneys start to secrete hydrogen, meaning move it from the blood into the filtrate and ultimately the urine, so we can get rid of that excess hydrogen in the urine, and reabsorb bicarbonate so we have more base driving the reaction in the opposite direction. This is a really important conceptual point to understand. The respiratory system manages pH by managing the levels of CO2, which influences this particular buffering system. The renal system influences pH by managing the amounts of hydrogen and bicarbonate directly. If the respiratory system breaks down, meaning the respiratory system is not working properly, it can result in pH disturbances like respiratory acidosis. So when the respiratory system isn't working properly, we say that we have renal compensation. In other words, if the respiratory system isn't working, the kidneys will kick into overdrive to try and maintain pH. On the flip token, if the kidneys aren't working or that acidosis is stemming from somewhere other than the respiratory system, we call it metabolic acidosis. And the system that's going to step up to try and handle some of that or to compensate for some of that is the respiratory system. Now, you're going to be asked to make one of 4 diagnoses. We have respiratory acidosis. What can cause respiratory acidosis? Anything that paralyzes the movement of air in and out of the lungs, anything that compromises the movement of air in and out of the lungs. So you can have lung diseases like COPD or cystic fibrosis or asthma, right? You could have paralyzed muscles due to something like ALS or an injury to the neck. Narcotic or barbiturate overdoses, right? Things like heroin or barbiturates can depress respiratory centers, and they can slow respiration way down, which is going to cause CO2 to build up, and it's going to produce a condition of acidosis. In other words, anything that compromises the function of the respiratory system will produce a type of acidosis called respiratory acidosis, right? Anything that increases the activity of the respiratory system and produces hyperventilation, where you're breathing too fast, like anxiety attacks, fear, panic, is going to suck excess CO2 out. And as CO2 is getting sucked out, that reaction is going to push in such a way that hydrogen ion concentrations go down, and that can produce clinically relevant respiratory alkalosis. So things that can cause respiratory alkalosis, panic attacks are a big one. High altitude is another big one for reasons I'm not going to talk about in a huge amount of detail. Anything that causes hyperventilation, essentially. One of the most common reasons for clinically relevant respiratory alkalosis is panic attacks. And it's also when people are undergoing surgery and the anesthesiologist calculates the rate at which ventilation should be occurring a little bit off. So the ventilator is pulling out, it's ventilating at too high of a rate, and it's pulling out CO2. That can cause respiratory alkalosis. Now, whereas you have respiratory acidosis and alkalosis, you also have metabolic acidosis and alkalosis. Metabolic acidosis and alkalosis will always in some way, shape, or form boil down to what's happening in the kidneys, right? So even if the cause is taking place somewhere other than the kidneys, if it's a metabolic form of the acidosis, it means that the kidneys aren't able to handle whatever's going on. So metabolic acidosis. Well, severe diarrhea can cause metabolic acidosis, but the big ones I want you to know, are diabetic ketoacidosis, because you're going to see DKA pretty frequently. So if the body can't burn sugar for energy, it burns fat. The byproducts of the breakdown of fat are ketone bodies. Ketone bodies can only be handled in the kidneys, so it puts a stress on the kidneys with respect to how they handle acid-base balance. That's a big one for metabolic acidosis. Diabetic ketoacidosis is a big one. And ultimately, we're going to look at how you diagnose those. So you can also have kidney disease or kidney failure, right? That's another big one. So diabetic ketoacidosis, kidney disease. Metabolic alkalosis, things like excessive vomiting. That's probably the most common cause of clinically relevant metabolic alkalosis, because the stomach is secreting more acid, meaning the alkaline tide becomes greater, and the kidneys need to handle that. Or an ingestion of excessive amounts of things like Tums, antacids. That's another really common cause for metabolic alkalosis. The bottom line is, in both of these, the kidneys are in some way, shape, or form involved. If the kidneys aren't doing their job or unable to do their job, the system that attempts to compensate is the respiratory system. If the respiratory system isn't doing its job, the system that attempts to compensate is the urinary system. And we refer to those as as the urinary system. So, types of cells found in the kidney. You have principal cells. Principal cells handle fluid and electrolyte balance. So the principal cells along the collecting duct of the kidney, and the collecting duct is a whoppingly important part of the nephron. Principal cells in the collecting duct have receptors for antidiuretic hormone. So when antidiuretic hormone binds to principal cells in the collecting duct, it triggers the insertion of aquaporins, and we get the reabsorption of water. When aldosterone binds to intracellular receptors, it activates gene expression through the process of transcription and translation. We produce more sodium-potassium pumps, more potassium and more sodium channels. Potassium gets secreted, right? Sodium chloride gets reabsorbed. With that salt comes water. You got salt and water reabsorption. So principal cells handle salt and water balance. There are a unique group of cells in the collecting duct of the kidney. So the collecting duct is composed of principal cells. It's also composed of intercalated cells. And there are two types of intercalated cells. There are type A intercalated cells, which secrete hydrogen and reabsorb bicarbonate. And there are type B intercalated cells, which secrete bicarbonate and reabsorb hydrogen, meaning these get activated under different metabolic conditions. So when you're thinking about fat insults to the body, you can have, or acid insults to the body, you can have all sorts of things that influence blood pH. Blood pH is between 7.35 and 7.45. Now, let's say that we have a condition of acidosis. In a condition of acidosis, hydrogen concentrations are always going to be high, and pH is always going to be low. Anything below 7.35 is a condition of acidosis. By definition, it is just a condition of acidosis. Now, if a condition of acidosis arises, right, the respiratory system handles it by increasing ventilatory rate. So you start to breathe faster in order to get rid of CO2. The kidneys handle that with the type A intercalated cells along the collecting duct. So here we have the collecting duct. There are principal cells, right, like this one here. And then there are type A intercalated cells. When the system becomes acidic, so when the system becomes acidic, too acidic, type A intercalated cells will start secreting hydrogen and reabsorbing bicarbonate. So they'll secrete hydrogen and reabsorb bicarbonate. When we secrete hydrogen, we remove hydrogen from the blood, which hopefully will bring that pH back up and reestablish homeostasis. When we reabsorb bicarbonate, that's like reabsorbing base. So we secrete hydrogen, reabsorb bicarbonate. In a condition of acidosis, type A intercalated cells are activated. In a condition of alkalosis, type B intercalated cells are activated. So let's say that hydrogen levels are too low. pH goes up above 7.45. If pH is above 7.45, by definition, you are looking at a condition of alkalosis. In a condition of alkalosis, type B intercalated cells secrete bicarbonate, and we eliminate that bicarbonate in the urine, and they reabsorb hydrogen in order to bring the concentration of hydrogen back up to reestablish that pH of 7.35 to 7.45. Right? So hydrogen goes up, pH goes down. Hopefully we bring it back down into our physiological, our reference ranges. So on the exam, you are going to be asked to distinguish between respiratory acidosis, respiratory alkalosis, metabolic acidosis, and metabolic alkalosis given lab values. All of the lab values that I give you, you're going to see active what's called compensation taking place. So when you are looking at a lab, the first lab value that you want to find, right, when you are looking at a lab is pH. If pH is below 7.35, you have a condition of acidosis. You've answered half the question right there. You said, looking at pH below 7.35, by definition, that's a condition of acidosis, meaning hydrogen ion concentrations are high, right, and pH is low. If pH is above 7.45, by definition, you are looking at a condition of alkalosis. First thing you do when you get some clinical lab values on the exam is you look at pH. Now let's say pH is low. You go, okay, that's a condition of acidosis. Now I have to figure out whether this is respiratory or metabolic. How do you distinguish between that? You look at CO2. So if pH is low, but CO2 is high, it means the lungs aren't effectively getting rid of CO2. So if pH is low and CO2 is high, it means the lungs are not effectively getting rid of CO2, and by definition, that would be a condition of respiratory acidosis. It would be a condition of respiratory acidosis. If the respiratory system is compromised due to whatever, COPD or asthma or obstruction or whatever it is, right? The system that kicks in is the urinary system. We say that it's metabolic compensation. So the urinary system, what's going to happen along the collecting duct is those type A intercalated cells are going to start secreting hydrogen and reabsorbing bicarbonate. So the next value you go to is bicarbonate. If CO2 is high, you know that's respiratory acidosis. Now, if the urinary system is compensating, it's going to be secreting hydrogen. So the pH of the urine is going to be decreasing because the concentration of hydrogen in the urine is going to go up. And we're going to be reabsorbing bicarbonate, right? So in this case, we get metabolic or renal compensation. And you're going to see if the kidneys are compensating in respiratory acidosis, the bicarbonate levels are going to be really, really high because the kidneys are trying to ditch as much hydrogen and reabsorb as much bicarbonate as they can in order to try and compensate for the activity of the lungs. Now, with acidosis, whether it's respiratory or metabolic, you're always going to get decreased level of consciousness, lethargy, fatigue, right? And the big danger is that proteins denature and the person slips into a coma and dies, regardless of what type of acidosis you're talking about. That's your major concern. So now let's say we look at pH and pH is low. And we go, okay, our next thing is we're going to look at CO2. Well, let's say CO2 is low. Let's say CO2 is low. You go, okay, well, if CO2 is low, it means the lungs are working. So now let me go to bicarbonate, because the kidneys regulate pH by regulating the levels of bicarbonate. So if the kidneys aren't doing their job, if pH is low and bicarbonate levels are low, it means that the kidneys aren't reabsorbing bicarbonate and secreting hydrogen like they should. And by definition, we consider that metabolic acidosis. There are two forms of metabolic acidosis or causes that I want you to know. Renal failure or kidney failure produces metabolic acidosis. Diabetic ketoacidosis is another big one. When you can't use sugar for energy, your body uses fats. The byproduct of the breakdown of fat are ketone bodies. Ketone bodies are super duper acidic, right? So you go low pH, low bicarbonate levels has to be metabolic. And you can think of a couple of causes. You can even maybe look at your blood glucose levels, because if your blood glucose levels are through the roof, that's a pretty strong indicator of diabetic ketoacidosis. Not to mention, if we have a metabolic acidosis, the system that's going to be compensating is the respiratory system. And it compensates by increasing the rate of ventilation. We start to hyperventilate to try and get rid of as much CO2 as we possibly can. So with metabolic acidosis, you're going to see CO2 levels are really, really low. And the person is going to be displaying what are called Kussmaul respirations, Kussmaul. And they're going to be going, I mean, they are not subtle when you see them. And that's the respiratory system going, oh man, we're becoming acidic, I need to compensate. Right? Kussmaul respirations are obvious. Again, you're going to get lethargy, fatigue, slipping in and out of consciousness, decreased level of consciousness, altered mental state, etc. So let's say you look at pH and pH is too high. It's above 7.45. By definition, you have a condition of alkalosis. So the next thing you look at is you go, okay, is this alkalosis stemming from the respiratory system or somewhere else? Is it respiratory or is it metabolic? So you go to CO2. If the body is alkaline and CO2 levels are low, It means you are breathing too fast for whatever reason. Either you're hyperventilating from something like a panic attack, or the ventilation machine that people have, the ventilator that you're on during something like a surgery, or if you're in a persistent vegetative state, it's set too high and you're sucking out too much CO2. Because if CO2 is low in a healthy system, right, what will happen is your respiratory rate will decrease in order to allow CO2 levels in the blood to increase. So if you're alkalinic, if you have a condition of alkalosis and CO2 levels are low, you know that's the respiratory system. Now, alkalosis presents the opposite of acidosis. Rather than being lethargic, fatigued, slipping in and out of consciousness, potentially going into a coma, you're going to be agitated, unable to sleep, hyper-aware, hyper-vigilant, right? Muscle twitches, uncomfortable, irritable, right? So alkalosis presents totally differently than acidosis at a patient level. If it's respiratory alkalosis, the system that is going to try and compensate are the kidneys. How do they do that? Well, type B intercalated cells are going to activate, and we're going to start secreting bicarbonate into the urine and reabsorbing hydrogen into the blood in order to try and bring that pH back down. On the flip token, if you have a condition of alkalosis and you see that bicarbonate levels are high, well, if the body is alkaline, the kidneys should respond to that by activating type B intercalated cells, and those type B intercalated cells should start getting rid of the bicarbonate and reabsorbing hydrogen to reestablish pH. If you're alkalinic and bicarbonate levels are high, right, by definition, you are suffering from what's called metabolic alkalosis. And metabolic alkalosis, the most common causes are excessive vomiting, because the alkaline tide from the stomach increases and the kidneys have to handle that, or excessive intake of things like Tums, right, antacids, which are super basic, and the kidneys have to handle that increase in base. How do they do it? They should be secreting bicarbonate. They should be getting rid of it. If you're alkalinic and bicarbonate levels are high, means that the kidneys aren't able to do their job properly. Like either they're overwhelmed by the amount of bicarbonate or they're not working properly. Those are the two most common causes of metabolic alkalosis, by the way, excessive vomiting and excessive intake of things like Tums. But at the end of the day, it boils down to what the kidneys are doing. So the kidneys, you go, okay, if the kidneys are not doing their job properly, how is the respiratory system going to attempt to compensate? In an alkaline condition, that's actually going to slow down respiration, and the person who's suffering from metabolic alkalosis is going to start breathing really slow, or hypoventilating, because their respiratory centers are going to be suppressed, because the respiratory system is trying to hold on to CO2 in order to push the reaction in the direction that will produce more free hydrogen. Let's look at this lab value. This is an actual lab. First thing you look at if you get a lab value like this on an exam, you go to pH. So 7.35 to 7.45 is our reference range. pH is 7.02. Anything below 7.35 is by definition acidosis. Acidosis, when pH drops. So we know hydrogen ion concentrations are high, pH is low, so you go, ooh, that's a condition of acidosis. What's the first thing I'm going to look at? I'm going to look at my CO2. Well, CO2 should be between 34 and 45 mmHg. It's at 10. So CO2 isn't high. In fact, CO2 is really, really low. The lungs are working fine. So then you go to bicarbonate and you go, okay, so if the body's acidic and the kidneys are doing their job, they should be reabsorbing bicarbonate and secreting hydrogen. The bicarbonate levels here are really low. That means the kidneys are not doing their job properly. The lungs are working fine. The kidneys are not doing their job properly. By definition, that is metabolic acidosis. So it's metabolic acidosis, and you go, well, what are some potential causes? Well, you have, diabetic ketoacidosis. You have kidney failure. The way you're going to distinguish between diabetic ketoacidosis and kidney failure is you're going to come down here to glucose levels. Now, whatever glucose is being measured in, this is just a different measurement. It's not milligrams per deciliter, it's millimoles per liter. But glucose is in reference range. That means that this problem is probably stemming from the kidneys. So you go, okay, it's metabolic acidosis, likely stemming from the kidneys because blood glucose levels are fine, right? Explain to me why. Well, bicarbonate levels are really low. It means that those type B intercalated cells are not reabsorbing bicarbonate, or type A intercalated cells are not reabsorbing bicarbonate and secreting hydrogen. They're not working properly, and that's why we have a condition of acidosis with decreased bicarbonate levels. It's probably a kidney issue because glucose levels are within reference range. The system that is attempting to compensate is the respiratory system. And the reason that CO2 levels are so low is because the person would be hyperventilating in order to try and get rid of as much CO2 as possible, because the respiratory system is desperately trying to bring that pH back up. And it's ditching as much CO2 as it possibly can to do that. That's why you see the low CO2 levels. Finally, I'm going to ask you, is the heart in trouble, right? Would this person's heart be in trouble? Because when you think about what happens during conditions of acidosis, the most dangerous thing that happens is you get an elevation in potassium in conjunction with the elevation of free hydrogen. So if you're thinking about cardiac problems, you come down to potassium. If potassium levels are elevated, it means the heart's in jeopardy. So you look at those potassium levels really carefully, because elevated potassium levels will prevent the heart from repolarizing. And there's our diagnosis, metabolic acidosis. You get all this right here. We got our couple of causes, but one of the things with any type of acidosis, particularly metabolic acidosis, that you want to pay attention to is hyperkalemia. That means excessive potassium levels. There are cells in the body, right, that aren't associated with any of the cells that we've talked about, that in conditions of acidosis will try to bring pH back up by sucking hydrogen into the cell, but in order to maintain electrical neutrality, they will pump potassium out of the cell. So when acidosis gets really bad, this system activates, and as a consequence of that, you can produce hyperkalemia. When potassium levels in the blood are above reference range or above a standard range, the organ that you are worried about is the heart, because elevated potassium levels will stop the heart. On the flip token, when alkalosis gets really bad, potassium gets sucked into the cell and hydrogen gets pumped out because we're trying to bring pH back down. That can produce hypokalemia. Hypokalemia is really dangerous because it will paralyze your skeletal muscles, right? Meaning they will perpetually be repolarizing, and it will create a gradient that paralyzes skeletal muscles. Okay, so finally, we're going to get into a different condition. So whenever you're looking at these labs, you go, what's the first thing I look at? I look at pH. In this case, it's 7.469. So that's a little bit above reference range. So by definition, we're looking at a condition of alkalosis. And now you're wondering where the alkalosis is stemming from. So the second value you always look for is carbon dioxide. Now, if alkalosis is stemming from the respiratory system, it means that you are hyperventilating in some way, either because of a panic attack or because a ventilator has been set too high on you. If you are hyperventilating in some way, it means your CO2 levels are going to go down. So if you ever see a condition of alkalosis, and then you look at CO2 levels and CO2 levels are low, by definition you are looking at what's called respiratory alkalosis. So this is a respiratory alkalosis, and then you need to start asking why. Why is this person hyperventilating, or what's happening with this individual? Another really strong indicator there is oxygen saturation. It's way high, right? Meaning not only are we effectively getting rid of CO2, but we exhaled to get rid of CO2, we inhaled to get oxygen. So those O2 sats are way high. It's not a bad thing. It's just that tells us it's respiratory in this case. Now, when you're wondering about compensation, you go, if the respiratory system, if you're hyperventilating, and that's what's producing the alkalosis, right? The urinary system needs to compensate. Now, when you see this value for bicarbonate here that's within reference ranges, it means the urinary system or the renal system hasn't started to carry out what's called metabolic compensation because the deviation from the reference range isn't that severe yet. But, so this would be uncompensated, but always on the exam I'm going to give you situations in which compensation is actually happening. So, when you think about oops. Sorry. When you think about how the urinary system is going to compensate for alkalosis, it's going to attempt to ditch bicarbonate and it's going to attempt to reabsorb hydrogen. So if the urinary system or your metabolic systems are compensating, you're going to see that the bicarbonate levels are really low, and that's because the kidneys are doing their job attempting to get rid of bicarbonate. So respiratory alkalosis, exact opposite, right? You get hyper excitability, you're irritable, you can't sleep. Really dangerous thing is if alkalosis gets too bad, it can produce hypokalemia. And in conditions of hypokalemia, your skeletal muscles will paralyze. When skeletal muscles like the diaphragm paralyze, that's really dangerous because you can't breathe and you can't move. So yeah, there we go. So think about those on the lab values, have questions ready for me, and we are good. Have a good day.